What fun. We get to spend some time talking about how we form urine. Yes, this section is all about how we form pee. We know that the overall function of the urinary system is to remove metabolic wastes from the circulatory system and then make sure they get out of the body. We also know that the microscopic functional unit of the urinary system are nephrons. So therefore, should be common sense that it is the nephron that is going to remove metabolic wastes from the circulatory system and get them off into the collecting ducts so they can get shipped out of the kidneys down to the urinary bladder and then be sent out of the body. The nephrons will also regulate water and electrolyte balance. Now, if you remember when we talked about the digestive system, the large intestine is also going to play a role in helping control water and electrolyte balance, because that is the last place in the digestive system where you can actually pull those compounds, molecules, ions out of the digestive tract and get them back into the circulatory system. Those will be moved off to the kidneys, and the kidney will actually have the final say as to how much water and how many electrolytes, or what the electrolyte concentration is in the blood. I also want you to remember that our old friend, the hypothalamus, which has multiple functions and is sort of like this little powerhouse of the central nervous system, will also play a role in regulating water and electrolyte balance. So that's kind of like the Wizard of Oz behind the curtain, pulling all the levers and the strings. Clearly, the end product of urine formation is urine. There are 3 major steps in urine formation, and you need to know them in order: glomerular filtration, tubular reabsorption, and then tubular secretion. And for the most part, and for the purposes of this course, they will happen in specific parts of the nephron. So let's look. Hello. Come on, here we go. You've already seen this image. Let's look at it again. The image on the left, that little sort of side picture, is of the glomerulus surrounded by the Bowman's capsule, so that is going to be the renal corpuscle. That is the location of glomerular filtration. You could probably have figured out that on your own because you have a glomerulus and one of the steps is glomerular filtration. So sure enough, that's where glomerular filtration will occur. So the renal corpuscle is the combination of the Bowman's capsule, or the glomerular capsule, and the glomerulus. And again, that is where the first step in urine formation occurs. Glomerular filtration occurs there. You'll remember the rest of the anatomy of the nephron. You will have, just past the Bowman's capsule, you will have the proximal convoluted tubule, which leads to the loop of Henle, which leads to the distal convoluted tubule, which leads to a collecting duct. You'll remember that the nephrons that are shown on the left are the cortical nephrons. They have a relatively short loop of Henle. The nephrons on the right are the juxtamedullary nephrons, and those nephrons will have nephron loops that are going to dip into the medulla, and those are the ones that are going to help play a significant role in the concentration of water and ions that remain in the kidney. So let's move on. Now, you will remember that the glomerulus is a ball of capillaries. It arises from an afferent arteriole, and it terminates in an efferent arteriole. So the afferent arteriole brings blood in that blood is going to do something in the glomerulus, and then the blood will exit out the efferent arteriole. Remember that the diameter of the afferent arteriole is greater than the diameter of the efferent arteriole. And you've already told me that when you have a big pipe being drained by a small pipe, there has to be a buildup of pressure in the middle, and in between those two pipes, arterioles, is the glomerulus. So sure enough, there's going to be a buildup of pressure in the glomerulus. Remember that there is a filtration membrane, and anything that is small enough, so water, small molecules, ions, anything that is small enough is going to get pushed out of the glomerulus across that filtration membrane. Anything that is big, which includes plasma proteins or other large molecules, like fats, will remain in the circulatory system. They're going to remain in the blood and get moved out through the efferent arteriole. They are just too big to get out. Remember that filtration is a passive mechanism. It does not require ATP. When you hear filtration, think passive mechanism. That would be a good thing to know. In fact, this entire page would be a really good thing to know. That's a hint. Now, what's the limitation of filtration? Well, here's the deal. It's not only the bad stuff that gets pushed out of the glomerulus, it's also the good stuff. So glucose, fatty acid, amino acids, vitamins, ions, anything that is small enough to get pushed out into the filtrate gets pushed out. The process is not selective. The only thing that it selects for, and I'm using that word in quotes, so select in air quotes, it selects for size. So that's really not a selection criteria. So when you think about filtration at the glomerular area within the glomerulus, the process is not selective. Anything that is small enough, whether you need it or not, is going to get pushed out of the blood and be part of the initial filtrate. So we know that the glomerular filtration process is going to occur in the renal corpuscle. You will remember that the glomerular capillaries are fenestrated. So they're going to filter dissolved materials and water from the blood. That filtrate will go into the Bowman's capsule. Remember that proteins are big. They're not going to get filtered out of the blood. Filtration is due to that buildup of pressure between the afferent arteriole and the efferent arteriole. I will not ask you those numbers on a test. Most of the fluid that is in the tubule, that gets moved into the tubule, will be reabsorbed. And we're going to talk about what's happening in the rest of the nephron as we go through the steps in urine formation. There is a thing called the glomerular filtration rate, GFR, for those in the know, and that is going to be directly affected by blood volume and therefore blood pressure. So the juxtaglomerular apparatus is also going to be involved. I absolutely positively want you to know that the GFR is directly affected by blood pressure and blood volume. So you now have this filtrate that's in the Bowman's capsule and is going to move into the proximal convoluted tubule. So about 65% of reabsorption of stuff that you need is going to occur in the proximal convoluted tubule. So about 2/3, roughly 2/3. I won't ask you that percentage, but you should know the majority of reabsorption is going to occur in the proximal convoluted tubule. Tubular reabsorption is the process through which substances are moved out of the glomerular filtrate through the walls of the renal tubule back into the blood in that nest or network of capillaries that surround the nephron, which are the peritubular capillaries. Most, again, 65%, is going to occur in the proximal convoluted tubule. About 15% is going to happen in the loop of Henle. The remainder is probably going to happen, about 10% actually, I think, happens in the distal convoluted tubule, and a tiny little bit is going to happen in the collecting duct. But the big player in terms of reabsorption is going to be the proximal convoluted tubule. So that those reabsorbed substances are going to move out of the lumen of the tubule through the simple cuboidal cells of the proximal convoluted tubule, and then get returned to the bloodstream through the peritubular capillaries. Let me think, is there anything else I want to cover on this? I don't think so. I think that's good. I think we're good to go. Now, we already have, some more info. Reabsorption of water is going to occur via osmosis. Now, this is kind of cool because the entry of the filtrate into the proximal convoluted tubule is almost directly opposite the point where those afferent and efferent arterioles enter the glomerulus or exit the glomerulus. We know that the absorbed substances are going to move through the wall of the renal tubule, simple cuboidal ET, I would know that, hint, hint. And we know that the proximal convoluted tubule absorbs nutrients, and then it actually releases them into the interstitial fluid around tubule, and then they get picked up by the peritubular capillaries. But that's a little extra step. You need to know that that material is going to move out of the tubule and find its way into the circulatory system via the peritubular capillaries. Now, here's a question for you. And if you've watched the videos that go hand in hand with this lecture, you'll probably already know the answer. Why is this step called tubular reabsorption? Why isn't it just absorption? We'll talk about it in class, unless you Google it.